Hi everybody, I am going to give you the directions for the allele frequency activity. These directions assume that you have already read through the background information on the worksheet that's on Schoology and watched the sickle cell anemia and heterozygous advantage video. If you haven't done that, you need to go back and do that first or this won't make sense. So in the directions, it says that you need 50 red beans, 50 white beans, and 5 containers and 1 coin. I've modified that a little. If you don't have beans, it's not a big deal. I'm using these colored glass beads. Some people put them in vases. I really don't know why I have so many around my house, but I'm going to use the big one as my big A allele in place of a red bean, and this clear one as my little A allele in place of a white bean. And if you remember, sickle cell anemia is a recessive disease, meaning you need to be homozygous recessive. So have 2 clear beans in order to have the disease. If you are heterozygous, then you won't have the disease. And obviously, if you're homozygous dominant, you won't have the disease either. Instead of my 5 containers, I have 2 containers and 1 piece of paper. So I have 1 container labeled gene pool, and I've put all 100 beans in the gene pool and mixed them up. I have 1 container labeled non-surviving alleles for alleles that don't make it to the next generation. And then I have a piece of paper with big A, so homozygous dominant, heterozygous, and homozygous recessive on it so that you can keep track. So that's instead of 5 containers. If you want to use 5 containers, use 5 containers. I find this a little easier just to move the beans around. And then I have 1 coin, just a quarter. Okay, so the first thing you want to do, and let me get the PowerPoint up here. The first thing you want to do is make your prediction of what do you think will happen to the frequencies of the big A and little a, so dominant and recessive alleles, as a result of the presence of malaria. So we are running this model in an area where there would be malaria. Do you think the frequency of the dominant allele will increase or decrease? What about the recessive allele, and why? And once you've made your predictions, then you can jump into the activity. So the activity says that you are going to simulate fertilization by picking out 2 alleles without looking. 2 alleles because every person has has 2 alleles. So you're not going to look because you don't want to introduce any bias into your sampling. So I got 2 white alleles, so that's homozygous recessive. So I'm going to go back here. And it now says that I need to flip a coin. It actually says another person will flip a coin. That's because in class we did this with partners. Obviously, you can flip your own coin. And if you flip heads, that means that person gets malaria. If you flip tails, that means they do not get malaria. Some of you will flip every time you get a new individual, but some of you will notice that there's actually a pattern. So if you are homozygous recessive, which is what I just got, I actually don't need to flip a coin at all because it doesn't matter. Somebody with sickle cell anemia isn't going to live long enough to reproduce anyway. And so whether they have malaria or not really doesn't matter. In both cases, they live for a brief time, and we're going to place them in the little a container. The same thing with heterozygous, right? This is what we're talking about. Sickle cell anemia has a heterozygous advantage, meaning if you are heterozygous, you don't have sickle cell anemia, but you are also resistant to the malaria parasite. Having the presence of the 1 little a allele does something to your blood cells so that the malaria parasite cannot invade. It can't infect you. And so you've kind of got the best of both worlds. You don't have sickle cell anemia, but you are resistant to malaria. So again, the coin flip doesn't matter. Just so you know, not everything has a heterozygous example. So like hair texture, if you have wavy hair, you're actually heterozygous. You have 1 curly allele and 1 straight allele. there's no advantage to that. So don't think that everything that's heterozygous has an advantage. But if you pick two big A alleles, so in my case, two blue beans, then you do need to flip because you don't have sickle cell disease. So as long as you stay malaria free, you will live. But if you get malaria, then you're going to die of malaria. So that's really when you need to flip. So I'm going to pick a couple more. Let's see, I've got, again, two clear. Now I've got heterozygous. So again, I can just put that in the heterozygous container. Again, two clear. And I'm just going to keep going until I get, and normally I would say don't look, but I want to show you how it works, two big A alleles. So now I'm going to flip my coin. And it is tails. Tails. So tails is no malaria. So I'm going to put it in the big A, big A. Had I flipped heads, I would have put it into the non-surviving alleles container. And so I'm just going to keep going here for a little bit. Okay, so here I got another big A, big A. I'm going to flip my coin again. And as you can see, this time I got heads. So I put it in my non-surviving alleles container. They didn't have sickle cell anemia, but they did get malaria. So I've now gone through all of my beans in my gene pool. It's empty. I've got a bunch of big A alleles in my non-surviving alleles container. That's from the ones that were homozygous dominant, but they did get malaria. And then I'm going to angle my computer screen. You can see I've got all of these in different rows based on whether they're homozygous recessive, heterozygous, or homozygous dominant. You don't need them in such nice, neat rows. I just did that. And so now the next thing in my directions are I am going to count up all of the surviving alleles from homozygous dominant and heterozygous. I'm not going to count the ones from homozygous recessive because they only live for a short time. So we're counting which ones survive long enough to reproduce. So I can actually take all my little A's from the homozygous recessive pile and put them into my non-surviving. Once I've counted them up, then I'm going to record them here. So I ended up with 36 big A alleles. And so that's all of my homozygous dominant plus then 1/2 of each heterozygous pair, right? I'm basically counting all my blues. And 24 little A alleles. Now, if I were just to go forth and put all of these back in my gene pool, which I'm going to do in a minute, what I would find is that I eventually, when I go on to the next round, now I'm going to have fewer and fewer and fewer alleles each round. So I want these to reproduce. So I am going to take half of the surviving alleles from each set and add those in. So I'm taking all of my surviving alleles and I'm putting them back into the gene pool. And then I'm taking half of the number. So I had 24 little A's, so I'm going to take 12 little A's out of my non-surviving alleles and put them into the gene pool, and then half of 36 for the big A's is 18. So I'm going to take 18 out of the non-surviving alleles and put them back in the gene pool. Now I have a new gene pool. It's got all of my surviving alleles from the last round. So the 60 from the surviving from either homozygous dominant that did not get malaria or heterozygous, plus the half reproduced. So I added in 12 more little A's, which I took out of the non-surviving alleles, and 16 more big A's. Just so you know, that actually got me up to 54 big A's. That's fine. I just had to go back into my master stash of little glass beads here and add 4 more. Sometimes you're going to need to do that. Sometimes you won't. It kind of depends on each round. Had I had an odd number, right? I got lucky that I had 36 and 24, so that divides easily in half. Had I had an odd number, it doesn't matter. I just take either the bigger of the two, like if I had 25, I could either do 12 or 13. It doesn't matter, just be consistent. And so then I start all over again for my second generation. In the end, what I'm going to do is find 4 other students to share data. Because as you know, if we're looking at statistical significance, the greater your N, the greater your sample size, the better your data is going to be. So when you're filling out this table down here for the totals, that is you plus all the alleles for each generation from the at least 4 students you've shared data with. I don't have that, but so I'll just put this in. So I've got 36 here. Your numbers will be much bigger, right? You'll have almost 5 times that if it's you plus 4 students. And then 24 here. And I highly recommend that you actually work with 4 other students instead of just do this 5 times. A, because it'll save you time, but B, because science is best when it's social, and even just sharing data, you're going to be seeing like, wow, you got 36, I got 12. I wonder if I did something wrong. And maybe you did and maybe you didn't. But that's why we always want to share data, so that if our data seems really different from everybody else's, we at least stop and we question it. It may not be wrong. Biology is kind of squishy and you get really anomalous results. But it does make us want to question when we're getting data that's different from everybody else. So then the total here would be 60, 36 plus 24. And then when we say frequency, we really mean percent. So in order to get, it's essentially saying 36 is what percent of 60. So 36 divided by 60 times 100 to get our percent is actually 60%. And then we would either do 24 divided by 60, or we could just say, well, I know it's got to add up to 100, right? So this has to be 40, right? Because if we're looking at percent, then altogether, it has to be 100%. 60 plus 40 is 100. And then I'm going to go ahead and graph that. When I graph it, I'm actually going to start at generation 0. Everybody has the same generation 0. Generation 0 is 50-50. So on your graph that you're going to make, you are going to have it start on the x-axis with just a dot at 50, because that would be 50 for big A and 50 for little a, and then those 2 lines are going to diverge. Please, if you have any questions while you're doing this lab, make sure you reach out so that we can help you. Have a good day.